A very good afternoon to Dr. Liu and to all my fellow friends. So today I'm going to present on the chloroplast DNA genetic markers. So the evolutionary history of chloroplast. Chloroplast undergoes secondary endosymbiosis after the primary endosymbiosis, which involves mitochondria, but on the secondary endosymbiosis, it involves the ingestion of cyanobacteria by the ancestral eukaryote cell. You can see here the ancestral eukaryote cell will ingest the cyanobacteria that contain the photosynthetic pigment that will evolve into chloroplast after a very long period of time. And then you can find the chloroplast in the modern eukaryote cell. The structure and function of chloroplast. So this is the structure of chloroplast cell. It involves the outer membrane here, the inner membrane here, the granum. Granum is the whole stack of thylakoid. Thylakoid is the one plate, something like this. Thylakoid. The lumen is the inside of the thylakoid. The stroma, the environment here, inside, and that's it. So the function of chloroplast is to capture the light energy and store it to be used in photosynthesis. So photosynthesis is the sugar production process in plants. So the genes that play an important role in the function of chloroplast and the gene function. So the PSBA is a gene that encodes for photosystem 2 protein D1, which uses light energy to abstract electrons from water, generate oxygen, and a proton gradient used in ATP formation. ATP is the energy that will be used in the photosynthesis process. And then the second gene is the PSBB. It is a gene that encodes for photosystem 2 CP47 protein in the chloroplast. And then the next gene is the METK gene. It codes for matureous-like protein involved in group 2 intron splicing in chloroplast. So genes in chloroplast, there are like almost 101 genes present in the chloroplast, but there are some that encodes for protein and there are some that does not encode for protein. So the gene that are protein coding is the PSBA gene, PSBB gene, and the METK gene, while the genes are not protein coding are the RBCL gene, PSAI gene, and RPL23 gene. So the METK and RBCL is the two most commonly used genes than any other genes in chloroplast because they are considered as the standard plant DNA barcoding markers due to their universality, relatively high overall sequence quality, low cost, and high discriminatory power between angiosperm. And this is all the reference that I use to for this presentation. Thank you.